So welcome back. This is going to be our screencast for chapter 24, and we are going to continue our discussion of evolution. But what we're going to do now is we're going to sort of step away from the mechanism of evolution. And in other words, we're not going to talk as much about natural selection. We're also going to sort of step away from the idea of how populations evolve, and that's what we had looked at back in chapter 23. Now what we want to do is we want to look at the origin of species. In other words, what we want to do is we want to look at how or why different species come to be. So a word we often use when we look at the idea of creating a new species is one called speciation. And what speciation does is, in terms of a concept, is it helps us to understand how microevolution and macroevolution are involved in creating those new species. Now, if you recall, microevolution was going to consist of adaptations or changes that actually evolve within a particular population. And typically, that microevolution was going to be confined to one gene pool. And that was a large part of our emphasis back in chapter 23 when we talked about changes in population. So that idea of changes in allele frequency, remember we had talked about Hardy-Weinberg, was important in sort of indicating whether or not there is a change in that population that might indicate a possible new species being formed. Now, the second type of evolution that we look at that oftentimes will help us sort of understand speciation is one called macroevolution. So the first one we looked at was micro, in other words, on a small scale. But when you look at macro, now we're looking at the large scale again. In other words, we're looking at not maybe the formation necessarily of a new species, but maybe the formation of a new group. But you got to understand is that sometimes the mechanics of speciation and those events that are involved to create that new species will often actually create large groups as well. Now, biologists, what they're going to do is they're going to compare different types of evidence to validate whether or not speciation is actually occurring. And so scientists today, what they'll do is they'll look at something called morphology. And morphology is simply the body form that they notice within a particular species of animal or plants. And they'll make comparisons between the body forms of that particular group and maybe the group of another animal or plant. They'll also look at physiology. Now, physiology is a little bit different from morphology because instead of looking at what you see on the outside, you're also going to take a peek as to what's on the inside. And in addition to looking at structure, you're also going to make observations of function. Now, biochemistry is going to be the third thing that a scientist might look at in terms of sort of identifying whether or not maybe two different organisms are separate species. And what they'll do is they'll look at the chemical processes that occur within each group. And then, of course, with the technology that we have today, DNA sequencing is also something that can be used and is often used to group organisms. Now, there's several different definitions that we could use for species, but the biological species concept is going to be the one that you most often see in biology textbooks. So the biological species concept simply states that a species is a group of populations whose members have the potential to interbreed. In other words, they can breed with one another. And they do this in nature, and what they do is when they do interbreed, they actually do produce viable and fertile offspring. They do not interbreed successfully with other populations, and if they can't interbreed with those populations, then we would consider them separate species. Now, when we look at that whole idea of barriers to the interbreeding between those different groups, oftentimes we talk about something called reproductive isolation. Now, reproductive isolation is the existence of what we would consider biological factors that actually impede or prevent two different species from producing those viable, fertile offspring. Now, if we do happen to have reproduction occur between two different species, if an offspring is produced, sometimes we'll use the term hybrid to describe those offspring. Now, reproductive isolation can be classified by whether factors will act either before or after fertilization. If they act actually before fertilization, we call that prezygotic. In other words, before the fertilization of the egg. So prezygotic barriers would block fertilization occurring, and it can actually happen by either impeding different species from attempting to mate. In other words, there's a reason why they really don't make the attempt to reproduce. It could be where they might make the attempt, but it actually prevents the successful completion of the mating. So the attempt is made, but mating is not successful. Or if mating is actually successful, then fertilization itself is going to be prevented. Now, prezygotic barriers maintain that reproductive isolation. And if you notice down here towards the bottom, there's going to be 5 different means of prezygotic barriers. One of those is temporal, the second is the habitat, the third is going to be behavioral, the fourth is going to be mechanical, and the fifth is going to involve gamete isolation. Now, the ones that we had talked about on the previous screen, those were considered prezygotic barriers. So remember, what that means is before fertilization of the egg. And the 5 that we had listed on the previous screen, one of them was habitat, 2 was temporal, 3 was behavioral, 4 was mechanical, and like we had said, that gametic isolation was going to be the 5th one. Now, when you talk about habitat isolation, basically you have a habitat that is going to it's not going to be able to be crossed by different species. And a good example of that would be the garter snakes that you see down here towards the bottom of the screen. If you notice, it's really apparent that we actually have 2 snakes that live in very different habitats. This one's going to live in an aquatic habitat, while this one is going to live more of a terrestrial lifestyle. Now, because of their different habitats, the likelihood of them coming across each other and actually attempting to reproduce are going to be rather rare, and it's all based on that habitat isolation. Now, temporal is going to be another situation where, because if you have individuals in different populations that actually reproduce possibly maybe in different times of the year, maybe different times of the day, that would be considered temporal isolation. Now, behavioral is going to be the third type, and that simply means that we have 2 individuals that even when they do get together, the behaviors that you observe during reproduction for each different species, they're going to be just different enough where one's not going to recognize the other's behavior as being reproductive in nature. So that's going to be behavioral isolation. The fourth is going to be mechanical. Now, mechanical basically means that we have 2 individuals, even though they look similar to each other, when you look at each species, because of the way they're put together, the parts that are necessary to actually reproduce basically aren't going to go together like they should. So in this case, they have 2 snails that they're using to sort of demonstrate this mechanical isolation. So the reproductive pores for each snail is going to be located in a different spot. Now, the reason it's located in a different spot is entirely because the spiral that you see on the shell of this snail is going in the opposite direction to the one that you see on this snail. And so what that does is it actually puts the reproductive pore on a different side of the animal. And because of that, when they come together, those pores cannot match up and reproduction cannot occur. And then we have something called gametic isolation. And gametic isolation basically means that the gametes that are being used to reproduce do not basically fit together. Now, as I had said, that's prezygotic barriers. The second type is a postzygotic barrier. Now, that's going to be after fertilization of the egg. Now, if you look down here, we actually have 3 different examples of postzygotic barriers. One of them is reduced hybrid viability. So what that simply means is that if you do have reproduction occur, maybe for example like you see here between 2 salamanders, that even though you do get offspring, the viability or the likelihood that those offspring will survive probably isn't going to be very great. In other words, the offspring that are produced are going to be much weaker than the parents. The second is reduced hybrid fertility, and a really good example of that is going to be the mule that you see right here. So if you notice, we have a cross between a donkey and a horse, and the cross that occurs between these 2 individuals, 2 different species, actually does produce offspring, but because it's a hybrid, the ability of that offspring to reproduce is probably non-existent because they're infertile. So the third one that we see here is something called hybrid breakdown. And what this simply means is that when these 2 individuals cross, there's a good chance you might actually produce fertile offspring in the beginning, but if those hybrids actually reproduce with each other, then the viability of the offspring becomes less and less and less. As I had said before, there's other definitions of species that oftentimes scientists will entertain. One of them is called morphological species concept, and this is going to be used to define a species by what we would consider measurable physical features. Now, the hard part about the morphological species concept is that a lot of times the information that is being used is subjective. And then a lot of times the information is coming from extinct animals. In other words, it's coming from fossils. Now, another definition of species that can sometimes be seen is one called the ecological species concept. And this is going to view a species in terms of its ecological niche. Now, you got to remember that a niche, and this is going to be something that we had looked at back in Honors Bio or Bio, is going to be the interaction of those individuals with both their living and non-living environment. And so when you make comparisons between different species, that ecological niche in this case becomes very important. In other words, maybe this one can live in this area, but because of the interactions it has with its environment, it's not going to be able to live maybe in the area of another species. Now down here towards the bottom, this is going to be considered the phylogenetic species concept, and this one basically defines a species as the smallest group of individuals on the phylogenetic tree that we would create based on its DNA or amino acid sequence. And so over here on the right, what you can see is this is an example of a phylogenetic tree. And if you notice, we have the different taxonomic classifications up here towards the top, and at the very bottom, which in this case is to the far right, is going to be the species that we're making comparisons of. And so in this case, as we said, when you look at this phylogenetic tree as a whole, what you either see on the bottom or in this case on the far right is going to be the smallest group. Now, speciation can take place with or without geographic separation. So it's really important to understand that oftentimes what we do is when we look at separate species being created, sometimes we sort of imagine that there needs to be some sort of very large geographic barrier between them, but that's not always the case. So speciation can occur in two ways. It could be allopatric or it can be sympatric. So allopatric speciation is where you do actually have a geographic barrier that's going to separate those populations. The second group is going to be called a sympatric speciation, and this is going to be the example where really there's no geographic barrier between those two groups, but new species do form. Now, down here, what you see is you basically see a good example that sort of illustrates the difference between allopatric and sympatric speciation. So this right here would be our original population, and over here on the left, this is going to represent that geographical barrier that has occurred in this particular type of allopatric or patric speciation. Over here on the right, this is going to be the sympatric speciation, and so maybe we don't have the geographic barrier, but because of some other reason, these two groups have sort of broken off from each other. In other words, they have developed into new species. Now, allopatric speciation, again, is going to be a speciation where we do have that geographical isolation. Now, when you have geographically isolated subpopulations, you need to understand that that gene flow between those populations is going to be reduced. And as we had said, it's going to be geographic, so you might be talking about things like mountains or maybe glaciers, or it could even be a water barrier between those subpopulations. Now, when you talk about allopatric speciation, separate populations still may evolve independently through the mutation or natural selection or the genetic drift that we had talked about in previous chapters. Now, the reproductive isolation between populations generally will increase as the distance between them also increases. And we have two examples down here that sort of illustrate that. So the whole idea of that adaptive radiation that we had talked about in previous chapters often will occur on long island chains. Now, isolation is going to cause the founders, in other words, those that were actually sort of broken off from that original population, it's going to cause them to undergo that speciation that we're referring to. Now, Darwin's finches is a really good example of this reproductive isolation in regards to adaptive radiation. Many of those diversely adapted species actually were formed from a common ancestor. And so in other words, they had broken off geographically from the original population, and based on what they had found on those islands in terms of maybe food, habitat, etc., they had developed into a brand new species. Now, sympatric speciation, as we had said, is going to be one where really there's no geographic isolation between the groups. And so when you talk about sympatric speciation, we talk about something called polyploidy. Now, polyploidy, if you look at the prefix, poly means many, is basically going to mean the presence of extra sets of chromosome due to accidents during cell division. So in this case, you may not necessarily have two species interacting, but what you end up with is maybe the creation of a new species simply because of an accident that has occurred during that cell division. So when you look at that polyploidy, we actually break that down into two categories. One is called autoploidy, and the other one is called alloploidy. So an autoploid individual is going to be one that actually has more than two chromosome sets. But those two chromosome sets have been derived from one species. So over here, if you look on the right, this is going to be a good example of an autoploid type of situation. If you remember back from the information you had looked at in terms of meiosis and mitosis, the 2N represents the diploid state in this particular cell. And so in this case, we have 6 chromosomes being represented. But because of a failure of the cell to divide after chromosome duplication, what it has done is it's actually given rise to a tetraploid type of tissue. So instead of having 6, now what we have is 12. So it's now considered tetraploid, in other words 4N, and not diploid or diploid. So then what happens is we continue, and if gametes are produced by this individual, then diploid gametes are being produced now instead of haploid gametes. And if those gametes come together, then the offspring that are produced are going to be tetraploid. Now, when we look at autoploid, autoploid is going to be the result of a failure of meiosis in most cases, but because of the situation and the numbers that are involved with an autoploid situation, they can self-pollinate or actually mate with other autoploids, but they cannot mate or reproduce with diploids. Now, the second group is going to be called alloploid. So this is going to be a species with multiple sets of chromosomes, but they have been derived or taken from different species. Now, in this case, the hybrid that's produced may be sterile, but there is a chance that it actually might propagate asexually. So again, over here on the right, this is going to be the example of alloploid. And if you notice, we have species B and we have species A down here towards the bottom. Meiosis has occurred for species A in a normal way, but there's been a meiotic error for species B. So if you notice, what has been produced is an unreduced gamete with 4 chromosomes. And if you notice down here, this is the normally reduced gamete. And so instead of having 6, now they have 3. But if these two gametes come together, again from 2 different species, what we end up with is we end up with a hybrid with 7 chromosomes. Now, if by chance that particular hybrid actually reproduces with a normal gamete, then if you notice over here on the far right, we get a viable fertile hybrid that's going to be diploid, but now have 10 chromosomes. Now, sympatric speciation can also occur from habitat differentiation, in other words, maybe changes within that habitat, and possibly sexual selection. So, sympatric speciation that occurs as a result of habitat differentiation basically involves the formation of new ecological niches within that habitat. And these are going to be ones that were not previously exploited by the parents. So a good example of that is going to be the maggot fly. The maggot fly is able to live on native hawthorn trees in North America. But because of our more recently introduced apple trees, the maggot flies have now found a new niche. In other words, they have a new place where they could actually live, feed, reproduce. And so now they can actually live in both areas. As we had said, the second one is going to be sexual selection. A really good example of this is going to be the cichlid fish that you would find in Lake Victoria. And if you look down here, the easiest way to understand this is by looking at these 2 diagrams. If you notice the one on the left, this is sort of gives you an idea as to how the color changes occur within these particular cichlid fish. And so what you notice is the ones that are towards the top, they are very sensitive to blue light. And because of this blue light sensitivity, the blue coloration tends to be favored. And so the fish that you'll find towards the upper portion of the lake are going to be blue in color. Those that were found more towards the bottom tend to be sensitive to red light. And so they tend to take on the red coloration. So what you end up with is you end up with 2 populations of fish. You end up with the cichlids towards the top being blue in color, the cichlids towards the bottom being red in color. Now, speciation occurs because any of the males that are found towards the bottom, if they tend to make their way towards the top of the lake, they're going to be the ones that are going to be unsuccessful with mating because females that you would find towards the top are going to prefer the male fish that are blue as opposed to the red fish that are down towards the bottom, and vice versa. So if we have any males towards the top that actually make their way towards the bottom, the females towards the bottom are going to prefer the red males over the blue males. So one of the big questions that often comes about in terms of speciation is that how long does it really take for a new species to come about? So what scientists have done is they've actually used various different types of data, as we had said. Fossil records might be one, morphological data could be another, and again, more currently, the molecular data that we had mentioned earlier. From the data that has been collected, what scientists have done is they've actually seen 2 different categories or forms of evidence that have shown how rapidly a new species can come to be. The first one is called rapid change. And so what that simply means is that we have a new species that for whatever reason has appeared suddenly. It sticks around for a period of time, but then sometimes it actually disappears almost as suddenly as it appeared. This is called a punctuated equilibrium model. Now, the problem with this model is that it contrasts with Darwin's idea of how a new species would come about, because in his idea, it takes a really long period of time to create a new species. And his idea was based on the idea of gradualism. So gradualism basically means a slow and continuous change over time in a species' existence. Now, this can be demonstrated down here towards the bottom. This one right here is going to demonstrate the idea of punctuated equilibrium. So if you notice, both types of equilibrium or patterns actually start off with the same type of butterfly. But if you notice for the punctuated example, we might have a butterfly that might produce this particular phenotype of offspring, but then for whatever reason, who knows, maybe through mutation, it produces this form of offspring. And this particular offspring could actually take advantage of maybe either that habitat or maybe another habitat. And because of that, it sticks around. Then down here towards the bottom, again, this is the same butterfly, but if you notice, the idea of change occurs in a very gradual way. So in other words, if you make comparisons between the two parts of the diagram, we still end up with a butterfly that is very similar in both cases, but the idea of that change occurring is not really sudden. It tends to change color gradually over that long period of time. All right, so that's going to finish up this screencast for Chapter 24, and as always, please make sure that you have completed your screencast study guide.